Hello Andy, this is your Ravindra, Senior Botany Lecturer. Welcome to our channel. Our channel name is Avedya's Edu Club. So in this chapter, now we are going to discussing about the one more important 8 marks question for IP examination point of view. That is Krebs cycle. Krebs cycle. So in the previous class, we have to already discussed about the glycolysis process. That is also most important 8 marks question for IP examination point of view. And in this chapter, it is also most important for IP examination point of view. Krebs cycle also 8 marks question. Okay. So Krebs cycle. Krebs cycle is also known as TCA cycle, tricarboxylic acid cycle. And why it is called as Krebs cycle? It means the scientist, Sir Hans Krebs scientist, proposed the complete pathway of the Krebs cycle. Hence it is called as Krebs cycle. It is also known as TCA cycle, tricarboxylic acid cycle. Why? Because in this Krebs cycle, the first compound is citric acid. Okay, citric acid is formation, that is the 3 carboxylic groups is present in the citric acid, hence it is called as tricarboxylic acid cycle. So in this Krebs cycle also 10 reaction is participated and 10 enzymes also participated. So I will tell you what is the 10 enzymes and 10 reactions in this Krebs cycle one by one. The 10 enzymes, 10 reactions. First one is condensation. Second one is dehydration. Third one is hydration. And fourth one is oxidation. And fifth one is decarboxylation. And sixth one is oxidative decarboxylation. And seventh one is cleavage. Eighth one is oxidation. And ninth one is hydration. 10th one is oxidation. So these 10 reactions is involved in the complete Krebs cycle. Okay, condensation, dehydration, hydration, oxidation, decarboxylation, oxidative decarboxylation, cleavage, oxidation, hydration, oxidation. So we have to remember here, first in this Krebs cycle, it is start with acetyl coenzyme A. So in the previous class, we have to already discuss about the glycolysis process. In that glycolysis, the end product is pyruvic acid. So pyruvic acid is the end product of the glycolysis, and it is the 3 carbon compound of glycolysis. So this pyruvic acid undergoes to the oxidative decarboxylation reaction. What is the reaction? Oxidative decarboxylation. Oxidative decarboxylation reaction to form the acetyl coenzyme A. So 3 carbon compound. It means in this oxidative decarboxylation reaction, one coenzyme A is added here, and one carbon is given out in the form of carbon dioxide. It is pyruvic acid now changed into acetyl coenzyme A. So 3 carbon compound of pyruvic acid now change into 2 carbon compound of acetyl coenzyme A. Pyruvic acid is the 3 carbon compound, acetyl coenzyme A is the 2 carbon compound. It means one carbon, from this 3 carbon, one carbon is lost in the form of carbon dioxide. So here the acetyl coenzyme is a 2 carbon compound. So this reaction is called as oxidative decarboxylation. So oxidative means here the redox reaction takes place. It means NAD, nicotinamide adenine dinucleotide, gets reduced to NADH2. And decarboxylation means next to one carbon is lost in the form of carbon dioxide and one coenzyme, it is the intermediate factor, is added into this reaction to form the acetyl coenzyme A. So it is a 2 carbon compound. Pyruvic acid is a 3 carbon compound. It means one carbon is lost in the form of carbon dioxide. So pyruvic acid is the end product of the glycolysis, and acetyl coenzyme A, it is the connecting link between the glycolysis as well as Krebs cycle. So acetyl coenzyme A is also known as, it is the connecting link between the glycolysis and Krebs cycle. So this Krebs cycle is start with acetyl coenzyme A. You have to remember this point. Okay, Krebs cycle. First start with acetyl coenzyme A. Acetyl coenzyme A. So acetyl coenzyme A, we know already, it is a 2 carbon compound. Acetyl coenzyme A undergoes to the first reaction, that is condensation. It means the acetyl coenzyme A, it is accepted to the oxaloacetic acid. OAA means oxaloacetic acid. So oxaloacetic acid is the 4 carbon compound. Acetyl coenzyme A is the 2 carbon compound. It is acetyl coenzyme A is condensed or accept to the oxaloacetic acid. So acetyl coenzyme A condense with oxaloacetic acid and to form the citric acid. Citric acid. Okay, acetyl coenzyme A. First Krebs cycle start with acetyl coenzyme A. It is a 2 carbon compound. It is condensed with oxaloacetic acid to form the citric acid. It is the first compound of Krebs cycle. So this Krebs cycle is also known as citric acid cycle. It is also known as TCA cycle. It means TCA means tricarboxylic acid cycle. Next to the Krebs cycle is also known as citric acid cycle. So this is the first reaction of the Krebs cycle. Okay. Next, citric acid. How many carbons here? 2 carbons. And the oxaloacetic acid, 4 carbons. So totally in the 6 citric acid, 6 carbons is there. Okay, 6 carbon compound of citric acid is formation. Next, so this citric acid undergoes to the second reaction. What is the second reaction? Dehydration. So D means remove. What is the remove? Hydration. It means removal of water. So here the water molecule is removed. Okay. Next, citric acid undergoes to the dehydration reaction to form the cis aconitic acid. Cis-aconitic acid. It is also 6 carbon compound. Next, so this is a second reaction of the Krebs cycle. Next, cis-aconitic acid undergoes to the third reaction. What is the third reaction? Hydration to form the isocitric acid. Isocitric acid. It is also 6 carbon compound. And hydration means water molecules. Addition of the water molecule is added in this reaction. This is the third reaction of the Krebs cycle. Next, isocitric acid undergoes to the fourth reaction. What is the fourth reaction? That is oxidation. So to form the oxalosuccinic acid. Oxalosuccinic acid. So oxidation means 6 carbon compound of isocitric acid undergoes to the oxidation reaction to form the oxalosuccinic acid. It means fourth reaction, that is oxidation. It means one redox reaction takes place. It means NAD, nicotinamide adenine dinucleotide, it gets reduced to NADH2, nicotinamide adenine dinucleotide. It is gets reduced to NADH2. So this is a oxidation reaction. This is a fourth reaction of the Krebs cycle. And how many carbons is there? Oxalosuccinic acid also 6 carbon compound. Now, oxalosuccinic acid undergoes to the, what is the fifth reaction? Decarboxylation to form the alpha ketoglutaric acid. Alpha ketoglutaric acid. So, we know already decarboxylation. D means remove. What is the remove? Carbon. So, from this 6 carbon, one carbon is lost in the form of carbon dioxide. Alpha ketoglutaric acid is the fifth carbon, 5 carbon compound. Okay, from the 6 carbon, one carbon is lost in the form of carbon dioxide, and alpha ketoglutaric acid is a 5 carbon compound. So this is the fifth reaction of the Krebs cycle. Next, alpha ketoglutaric acid next undergoes to the, what is the next reaction? Oxidative decarboxylation. Here the two reactions is participated, oxidation and decarboxylation. Oxidation means same, NAD. NAD gets reduced to NADH2. Okay, next decarboxylation means one carbon is lost in the form of carbon dioxide from the 5 carbon. From this 5 carbon, one carbon is lost in the form of carbon dioxide, and coenzyme A is added here. One intermediate factor is added to this reaction, and next to form the succinyl coenzyme A is formed. Okay, from these fifth to 5 carbons, one carbon is lost in the form of carbon dioxide. How many carbons is there? 4 carbons. Succinyl coenzyme A is the 4 carbons. Okay, this is the sixth reaction of the Krebs cycle. Next, so I will write this point here, oxidative decarboxylation. Okay, next succinyl coenzyme A next undergoes to the, what is the next reaction? Cleavage. We know already about this word. Cleavage means breakdown. Succinyl coenzyme A undergoes to the cleavage. It is breakdown into coenzyme A and succinic acid. Okay, succinyl coenzyme is a 4 carbon compound. It is breakdown into coenzyme A and succinic acid. So succinyl coenzyme A is changed into simple succinic acid. So in this reaction, some energy is released. So this energy is used to convert the GDP. Next to it is converted to GTP. Okay, succinyl coenzyme A breakdown into coenzyme A and it is changed into simple succinic acid. In this reaction, some energy is utilized. So this energy is used in this reaction. This energy is used to convert the GDP change into GTP. Okay, so this GTP ultimately gives us 1 ATP. Okay, so this is a seventh reaction of the Krebs cycle. Next, succinic acid. Succinic acid, how many carbons? 4 carbons. Same, 4 carbons. Next, succinic acid undergoes to the next reaction, oxidation, to form the fumaric acid. It is also 4 carbon compound. But in this reaction, oxidation means redox reaction takes place. But instead of the NAD, here the FAD is participated. FAD is changed into FADH2. FAD means flavin adenine dinucleotide. NAD abbreviation nicotinamide adenine dinucleotide. FAD abbreviation flavin adenine dinucleotide. Okay, in this eighth reaction, FAD change into FADH2. Instead of the NAD, here the FAD will be participated. Next, ninth reaction. Fumaric acid undergoes to the next ninth reaction. What is the ninth reaction? Hydration, and to form the malic acid. Malic acid also 4 carbon compound. Okay, hydration means addition of the water. Okay, this is the ninth reaction of the Krebs cycle. Next, last reaction is oxidation. Malic acid also next undergoes to the oxidation to form the same oxaloacetic acid. 4 carbon compound, oxaloacetic acid also 4 carbon compound. So in this reaction, NAD, nicotinamide adenine dinucleotide, gets reduced to NADH2. Okay, so this is the 10th reaction of the Krebs cycle. Okay, condensation, dehydration, hydration, oxidation, decarboxylation, oxidative decarboxylation, cleavage, oxidation, hydration, oxidation. So these are the 10 reactions of the Krebs cycle. So let us count the ATPs in this Krebs cycle. So here observe carefully. So how many NADH2 we are getting in this Krebs cycle? So observe carefully. Here the one NADH2, and here also one NADH2, and here also one NADH2. Totally 3 NADH2. So we get 3 NADH2 from one cycle of the Krebs cycle. But we have to remember here very well careful. So this Krebs cycle is takes place in 2 times. Okay, so which we will take 2 times. So in this one Krebs cycle we will get one NADH2 here, one NADH2 here, one NADH2, totally 3 NADH2. And how many FADH2? 2. Sorry, 1 FADH2. And how many ATPs? 1 ATPs. So in the previous class I have already told you, from 1 NADH2 totally 3 ATPs released. From these 3 ATPs, how many ATPs is next released from this 3 NADH2? Totally 9 ATPs. Okay, next from 1 FADH2, we have to remember here, from 1 NADH2 3 ATPs released, from 1 FADH2 2 ATPs released. 2 ATPs, it is a worth, and next 2 ATPs released, and next one here also 1 ATP. So totally 12 ATPs released from the one Krebs cycle. But this Krebs cycle takes place in 2 times. From these 2 times, each one is double. So 12 is next double. So totally next another one Krebs cycle. From one Krebs cycle, totally we will get 12 ATPs. Next 2 Krebs cycles. 2 Krebs cycles we will get totally 24 ATPs. Okay, 24 ATPs here. So this is about the Krebs cycle. So next I will tell you what is the 10 enzymes in this Krebs cycle. First enzyme. So acetyl coenzyme A condenses with oxaloacetic acid to form the citric acid. That enzyme is citric synthetase. Citric synthetase. Second enzyme name is aconitase. And third enzyme name also same, aconitase. And okay, citric synthetase, aconitase. And third next fourth enzyme name isocitric dehydrogenase. Isocitric dehydrogenase. And fifth one enzyme So fifth one enzyme is oxalosuccinic. Oxalosuccinic decarboxylase. Oxalosuccinic decarboxylase. And sixth enzyme name is alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. And seventh one enzyme name succinyl thiokinase. Succinyl thiokinase. Next eighth enzyme name succinic dehydrogenase. Succinic dehydrogenase. Next ninth enzyme name is fumarase. And tenth enzyme name is malic dehydrogenase. Malic dehydrogenase. Okay. First enzyme citric synthetase, aconitase, isocitric dehydrogenase, oxalosuccinic decarboxylase, alpha ketoglutarate dehydrogenase, succinyl thiokinase, succinic dehydrogenase, fumarase, malic dehydrogenase. So in this respiration in plants chapter, 8 marks question is most important, that is glycolysis and Krebs cycle. These two questions are most important for IPA examination point of view. Okay, thank you for watching. Please subscribe my channel.